The overall goal is to determine the effect of experimental alterations on the cell extension repertoire and how it relates to cell adhesion function in cancer.
This method can help identify key roles of the cell membrane extension repertoire in modulating cell behaviors such as cell adhesion, motility, and invasion. The main advantage of this technique is that it is relatively simple to perform and can be easily adapted to various experimental conditions.
The implications of this technique extend toward therapy or diagnosis of locally advanced or metastatic cancers because it helps to accurately quantify changes in cell extension repertoire associated with alterations in tumor cell adhesive and invasive capacities. It can also be applied to other systems such as ion channel regulation, which is known to play an important role in cell extension formation and normal physiological processes. Visual demonstration and recording of this method is critical as the counting of cell extension steps are difficult to learn because of the subtleties of their appearance at times.
A day before imaging, count SW13, SW-Neo, and SW-DKK3 cells grown in a 37 degrees Celsius and 5% carbon dioxide incubator using a hemocytometer. Place sterile glass cover slips into individual wells of 3 6-well plates. Use a separate plate for each cell line and plate the 3 cell lines at a density of 5,000 cells per well into these 6-well plates. Place the cells at an incubator at 37 degrees Celsius and 5% carbon dioxide overnight. The next day, from each well, aspirate the growth medium.